Mm-hmm.
איפה? תני לי את הטלפון.
שנייה.
אוקיי, זהו, בסדר. למה אני לא...
יש טלפון. יש לך את הטלפון שלי.
אוקיי. Dear colleagues, good afternoon to our Chinese participants and good morning to our Israeli participants. I, Professor Itamar Wilner, member of the Israel Academy of Sciences and Humanities, and Professor Yuliang Zhao, member of the Chinese Academy of Sciences, act as co-chairs of this bilateral webinar series on nanotechnology. The Israel Academy of Sciences and the Chinese Academy have long-term collaborations and that are reflected by many mutual scientific meetings in various fields, exchange of scientists, students, and most importantly, excellent friendship. Israel praises the scientific advances promoted by the Chinese Academy of Science and the activities of science in China. 2 years ago, we decided to host a bilateral meeting on nanotechnology between the Chinese Academy of Sciences and the Israel Academy of Sciences. Unfortunately, due to the corona pandemic, several delays in the organization of a frontal meeting occurred, and we decided to realize this effort by conducting a series of bilateral webinars in several topics of nanotechnology. Due to the time differences between Israel and China, we were forced to limit each of the webinars to 4 lectures and appropriate following discussions. The 4 webinars were defined to address the following topics: nano devices and nanophotonics, energy and environment, self-assembly and biomaterials, and nanomedicine and nanobiology. We had already 2 of these webinars, and today we have the 3rd webinar on self-assembly self-assembly and biomaterials. I realize that many scientists from both countries are disappointed for not being able to present their results and their research activities, yet I hope that all of us will understand the time limitations that I emphasized. During the organization of these webinars, we had to overcome many obstacles, and while organizing this webinar series. And I would like to thank our partners from China, Professor Zhao and Ms. Zhao from the Chinese Academy of Sciences for their efforts. I would like also to thank and mention the help and the tremendous help of the staff of the Israel Academy of Sciences. And I would like to mention Ms. Galia Finzi, the Director of Academy of Sciences, Joe Cohen, Director of the International Relations, Naama Shiloni, the Head of Public Relations, Yaakov Rotman, who is responsible at the Academy on the Computer Unit, and the technical efforts of Mr. Arik Futterman that we have seen already earlier while we prepared our presentations. I would like to thank particularly Dr. Yael Ben Chaim, who dedicated herself to organize these webinars, and she had sleepless nights in organizing these events. On behalf of Professor Nili Cohen, the President of the Israel Academy of Sciences, I am delighted to open the webinar series, and I wish all of you a stimulating and successful scientific event in this 3rd webinar. Unfortunately, Professor Zhao, my co-chair, due to other commitments, cannot welcome the participants, and as co-chair of the meeting today, we have Professor Tang as a representative. I would like and honored, I'm honored to invite Professor Tang, the Deputy Director of the National Center for Sciences and Technology at the Chinese Academy, to present his welcome. Please, Mr. Tang.
Thanks, Professor Itamar Werner. Dear Israel colleagues, on behalf of Professor Yuliang Zhao, I would like to welcome everyone to join the Israel-China Bilateral Series in Nanotechnology. China and Israel have made vast achievements in practical collaborations in the fields of nanoscience and nanotechnology. The platform construction has been pushed continually, and friendly exchange have been intensified consistently. The collaboration has displayed fruitful highlights in talent cultivation, bilateral meetings, and joint project application. The Israel-China Bilateral series in nanotechnology mainly focus on the latest progress and future development trend of nanoscience and technology, especially the preparation, characterization, application, and development trend of various nanomaterials, nanostructures, and related nanodevices. It provided a high-level exchange platform for the scientists in the field of nanotechnology and laid a solid foundations for long-term exchanges and collaborations in the field of nanotechnology between China and Israel. I hope that such collaboration will provide an outstanding opportunity for academic exchange, technical innovation, discussion on the future prospect, exchange of the people, and share our research policy as well as the resources. Finally, I wish a great success of this conference. Thank you.
Well, I guess it's my turn. It's a great pleasure to be to co-chair this session and to co-chair it with Professor Wilner and our moderator, Professor Galia Blum. I will introduce at the beginning of each lecture of the two Israeli lectures, the speakers. At the end of each lecture, which will be of 25 minutes punctlich, and at the end, Galia will take over and will moderate a 5 minutes discussion questions to the speaker. At the end, we will have a panel of all the participants, all the chairpersons discussing the future, comments, questions, etc. Let me introduce the first speaker, Professor Yuval Garini. Professor Garini was a professor at Delft University until 2007. This year, in 2007, he moved to my university, Bar-Ilan University, and was a part of the physics department. In particular, as an ordinator for him, he was the head of the Nano Center at Bar-Ilan University for 4 years. In 2020, he decided that Bar-Ilan University is indeed very far from him to drive from his home. So he moved to the Technion, where he is right now a professor of bioengineering materials or biomaterials. And well, that's what I can add. Now, I should say something about the topic of his research. He is very much interested in the interaction between DNA and proteins. And for this, he uses a large variety of tools, especially physical tools, spectroscopical tools. And I would like to give the stage to Professor Yuval Garini.
Thank you very much, Professor Gidanken, Professor Wilner, and Professor Jiang from the NCNST. It is really great pleasure and honor being able to give this lecture. And nanotechnology is very close to me, as well as the connection with our Chinese partners that goes on for already a couple of years. And I hope to be able to join the next China Nano Conference that is going to take place in August. And I will do all possible efforts in order to come physically and not only by a webinar to join the conference. Let me please share my presentation. I will describe my work on DNA origami as a tool for measuring the conformation of single-stranded DNA. just to explain and to give a very brief introduction of why am I interested in single-stranded DNA. So what we see here is a picture that emphasizes what's called the central dogma in biology. What the central dogma in biology says is that in eukaryotic cells, all the DNA is concentrated in the nucleus of the cell. And when a gene has to synthesize a protein, it happens in two steps. The first one is the cell makes a copy of the DNA, which is called messenger RNA, mRNA. The messenger RNA leaves the nucleus, goes outside from the nucleus envelope to the cytoplasm, to the main volume of the cell, where he arrives to the structure which is called the ribosome. The ribosome catches the messenger RNA, pulls it over slowly along the ribosome, and builds, according to the code, a line or a chain of amino acids. This chain of amino acids is the protein. Once the process is finished, it is folded to the relevant structure, and the protein is ready to go to do its job wherever it's necessary. Now, this is a very complex process, and we have to remember that the DNA, which is inside the nucleus, has approximately length of 3 meters. And this is packed in the nucleus that has a diameter of maybe 10 or maybe 20 micrometers. This is a very complex packaging problem. And it's not a packaging just for storage, because the DNA inside the nucleus is constantly working. It's constantly opening, being replicated, etc. So there is a very big question: how can it be stored inside the nucleus in a way that allows it to work on one hand and doesn't make it too messy from the other hand? And what you can see here is the nucleus, and you can see those chains in different colors. These are simulations that we made to show how a chromosome, one part of the DNA genome, what the conformation of that will be if there will be no constraints at all to the polymer, to the DNA. And as you can see, none of the chromosomes would like to stay inside the nucleus. The conformation that they would like to have is much larger than the actual volume of the nucleus. So it's clear that once the nucleus is enforcing the chromosome to stay inside the nucleus, there must be some mechanism that allows them to do that in a way that they would not get mixed with each other. It's not a trivial problem. We were studying that for some time, mainly by measuring live cells and following the dynamics of different points along the chromatin, along the DNA. You can see those spots that are moving in 3 dimensions, and then we can analyze their motion. And based on that, it's not the main topic, but we arrived to the conclusion that there must be not just chromatin and DNA, which is loosely moving inside the nucleus like a spaghetti ball, but that there must be something that we call chromatin network. And that network is most likely mediated by a protein which is called lamin A. And without that lamin A, we showed that everything becomes completely messy. Nevertheless, that's not the whole story, because we know that inside the nucleus, during different processes, for example, during transcription, when the messenger RNA, as you can see here, is being synthesized from the DNA itself, from the gene, or during replication, when the DNA has to copy itself before the cell is dividing, you can see that there are bubbles that are formed in the DNA, which means the DNA, the double-stranded DNA opens up, and we get points where there is a single-stranded DNA. Now, single-stranded DNA can critically affect the structure of the DNA inside the nucleus, because it makes points which are much more fragile and can fold in different angles much easier than the actual double-stranded DNA, which is more rigid. And that's why we decided in this study that I'm showing to study the conformation of the single-stranded DNA. We know, of course, a lot from polymer physics on the structure of DNA. We can model it by different models. In this case, what you see here is called FJC. It's a very simple model of a freely joined structure where you can simulate the polymer as if it is made from rigid rods that are connected by joints that are completely flexible. And once you do that, or using a different model like worm-like chain, you can easily know what's going to be the conformation and many properties of that polymer. However, and we know it very well for double-stranded DNA. And one of the main parameters is called the persistence length, which is more or less the size of the Kuhn length, the size of a rod that I showed before. Nevertheless, in single-stranded DNA, because it is much shorter, the problem is much more complex. There are some theories like what's called the OSF, named after Odijk, Skolnick, and Fixman, that predicted what should be what's called the persistence length, the main unit that determines the rigidity of single-stranded DNA. But as you can see here, it is not that simple to predict because it depends on a couple of physical or chemical parameters that are not exactly known. And therefore, you cannot really predict what's going to be the persistent length just by looking at the theory, because it is, as I said, it depends on too many parameters that we don't know exactly. So what is the persistence length of single-stranded DNA? We know that for double-stranded DNA, it is approximately 50 nanometers, but we don't exactly know what it is for single-stranded DNA. On top of that, when we are analyzing a single-stranded DNA polymer, like we can see here, there is another complexity that is being added, and that's the fact that because it's a single-stranded, if there are matching nucleic acids along the DNA, they may form what's called a stem loop, as you can see here. And it turns out that there are many possible stem loops that may form along the DNA. And if that happens, the practical or the actual total length of the DNA is not the nominal length of the single-stranded DNA, it is only the nominal length of the parts which are not taking part in forming stem loops. And that's a complexity that must be taken into account when calculating or measuring the single-stranded DNA persistence length. There are even theories, like the one I'm showing here by Yofe from the Hebrew University, that predicted that the persistence, that what's called the end-to-end distance of the polymer, you can see, for example, here, the end-to-end distance. One prediction said it should be 0, almost. Okay, why? Because there are so many stem loops that are formed along the polymer that finally the two ends will become almost next to each other. So there is a way to test it, but that's not the only way to test it. Fortunately, there are programs that are allowing to predict what are the stem loops going to be for a given sequence of the single-stranded DNA. One of them is called mfold, which is what we used. And as an example, you can see for the single-stranded DNA that we worked with, in this case, it's a 500 nucleotide single-stranded DNA. You can see the prediction of what should be the stem loops, assuming that all of them will be formed. There is another complexity that most likely not all the stem loops that are possible will, in practice, in reality, be formed. Some of them will, and some of them will not. We took that into account as well. Now, the single-stranded DNA persistence length was already measured before in different works. You can see here a couple of references where people tried to measure it. However, it was quite limited in the sense that in order to prevent those stem loops, those works were very limited either to measurement where the DNA was stretched by applying force on the two ends of the DNA in order to prevent the stem loops. But when you do that, of course, that it also affects the persistence length, or by other means, like for example, increasing the salt concentration to an extremely high level, which is beyond the natural salt concentration inside the cells in order to prevent those stem loops. And nevertheless, if you pick out the values that were measured, you can see a very high variability in the values that were predicted in the literature from those works. So we decided to adopt another method, which is based on DNA origami. I believe that most of the people here are already familiar with DNA origami. It's an amazingly interesting and I would say easy manner in order to construct two-dimensional or even three-dimensional nanostructures. You can see this from Rothmund's very famous paper in 1993, where he designed and synthesized many different types of structures. It can even be done from RNA itself, and it can even be done for structures that are dynamic. So the imagination can really go on for many capabilities that DNA origami can be used, even for drug delivery and for other applications. And it's a very active field of research. I'm actually familiar with quite a few researchers at the National Center of Nanoscience and Technology in Beijing that are, I can say, world leaders in the field. So we designed this structure that you can see here. It is made from a long single-stranded DNA, where here it goes back and forth on itself in two positions, one here and the other here. And these two are completely interconnected. So it leads to the formation of two rods that are completely fully rigid, but in between them, there is a piece which is only single-stranded. And we designed that structure. It's not extremely complex to do. We took advantage of another program that is freely available, which is called CAD Nano, that was developed in a collaboration between MIT and Harvard. And we designed it, we developed it, not very complex to build it. We based everything on a plasmid, which is approximately 7,250 nucleotides. Each one of the rods consists of 2,096 bases, and the remaining 3,000 nucleic acids are divided between the single-stranded here and the tails, which are not important for us, but it gives us a degree of freedom to make devices with different lengths of single-stranded DNA in the middle. The procedure of structuring, once you have the actual plasmid and what's called the staples, a very long list of short DNA, single-stranded DNA pieces, is not very complex. You mix everything together with the right salt concentration, pH level, and you run a PCR temperature cycle that basically starts from a high temperature that slowly goes down based on different parameters that I'll not enter to the fine details. And if everything works well, you get your structure at the end. And this is a more realistic scheme of the structure that we designed. You can see the two rods and the single-stranded DNA in between. And the purpose of the rods, the purpose of this structure was to allow us to identify very clearly the two endpoints of the single-stranded DNA. Once you can measure the endpoint distance of the single-stranded DNA, you can estimate the persistence length. Okay, so in some way, the main purpose of the DNA origami was to identify those two endpoints in a completely clear manner. And we did that. We measured many of those structures using an atomic force microscope. You can see here an example. So you can see these are the two rods, and you can see here the single-stranded DNA folded in some way that is not that easy to identify. Everything was done in the by using magnesium chloride at a certain concentration and very regular buffer for the solution. Basically, we just drop a drop of the structure on a mica structure and let it adhere to the surface and then make the measurement. You can see many structures that were identified. However, some of them are too complex because you don't exactly know what's going on there. So we isolated and we only looked at those that are clearly isolated from all the others. You can see them here below. And then there is no way to confuse the single-stranded DNA of any two structures. And we have many structures to select from. So once we have those structures, we can really form a measurement. So you can see the structure. And just for verifying that the structure is correct, we measured the length of the rods, which is supposed to be 100 nanometer. We measured the angle just in order to make sure that there is no specific angle. If there is any, it means that something is wrong in the random process of how the structure adheres to the surface. And then we measured the end-to-end distance. We also measured the width. So you can see, for example, the distribution of the rods. It is really very close to 100 nanometer as we predicted. The width of the rod is about 8 nanometer, also what we predicted. You can see the distribution of the angles that I showed before, very close to a uniform distribution, almost with no specific angle, which shows that the distribution is really random, which is good. And then, of course, we measured the distribution of the end-to-end distance. We had 4 different structures that has a different number of nucleotides, so a different length. You can see all of them. And then what we did, we know exactly what should be this distribution in 2 dimensions. It should be a Rayleigh function according to the persistence length after getting rid of the parts that are taking part in the stem loops. And you can see here the best fit of the Rayleigh function for each one of those structures. We repeated that calculation and fit after assuming that the total number of stem loops is different. And you can see in this summary the results. So you can see for the 1, 2, 3, 4 of the structures, you can see what is the predicted persistence length, assuming that either there are no loops completely, no stem loops in the structure, or that only 50% of the stem loops are really exist along the single-stranded DNA polymer, or all of them exist. So of course that according to each of those assumptions, we get a different value for the persistence length, but you can see that it all sums up to something which is quite on average on the scale of very close to 2 nanometer DNA. This was published some time ago in Nanoletters, so all the information can be found there. However, after doing that, we were a little bit worried that the rods themselves may alter the random distribution of the single-stranded DNA, because after all, these are relatively large structures that may stretch or may bias the natural end-to-end distance of the single-stranded DNA toward some direction, whatever this direction may be. So in order to overcome that, we decided to do another set of experiments, and this time to design a ring single-stranded DNA. And this time we designed a structure which is rectangular, as you can see here. And then there is a ring single-stranded DNA that is connected to that structure at the edge. Again, the purpose of the structure is in order to be sure that what we are looking at is only one single-stranded DNA. For example, when there is such a case, we neglected it. If we would only look at single-stranded DNA without the structure, then it will be difficult to tell whether we are looking at a single ring or maybe 2 or 3 that are interconnected or superimposed on one another. So having the structure prevents that complexity, and we can ensure that we have only a single stranded in each case. Again, we know the theory. This time, we adopted the method of comparing what's called the gyration radius. Roughly speaking, I can say that the gyration radius gives the radius of a circle that the polymer can fit into that. It's not exactly, you can see here the exact definition of that, but what I said is more or less the physical meaning of that. We know well what should be the average of the gyration radius for an ideal polymer. You can see it here based on the nominal length and the persistence length of it. The point is that we don't have an exact equation for the distribution of the gyration radius. And of course, that when you do an experiment, you work on many structures, you get a distribution. So what we did here, first of all, again, we calculated the stem loops. You can see that there are many stem loops formed along the single-stranded ring DNA in this case. You can see the zoom in. For some of them, it's clear why they are formed. And you can see that, roughly speaking, there's more or less 50% of the DNA that take part in it. So it must be taken into account for the calculation. You can see here as an example, one of the structures. You can actually see, even from the picture, that the structure is quite nicely designed. There are some missing points along the structure, but that happens. So this is a zoom in just to the single-stranded ring polymer and the way it is shown with the atomic force microscope. And when we look at along different points here, we can see that there is a very reasonable distribution of the DNA along those structures, and it can be measured. So the gyration radius can be calculated from what we see here. And for three different structures that each one has a different number of nucleotides along the ring chromosomes, we calculated the distribution of the gyration radius. You can see that it makes sense that it becomes shorter as long as the number of nucleotides is shorter. And now the next point is how to fit it. So we adopted the method where we did a simulation of the single-stranded DNA. We did it in 3 dimensions, but then took only the 2-dimensional projection of the 3-dimensional structure. This is what the DNA is supposed to look like on the structure. There is a lot of literature about it if someone will be interested. Now, the simulation that we did takes into account self-avoidance. So the polymer doesn't repeat itself at any junction point. And it also takes into account the stem loops and doesn't count them along the loop, but it does count them for the calculation of the gyration radius. And in order to fit the simulation to the measurement, we adopted the non-parametric data fitting of the cumulative distribution function based on the Kolmogorov-Smirnov test. And you can see here in red the CDF of the simulation, and in blue the CDF of the measurement. It's actually an excellent method for doing such a non-parametric fit. And then we tested the fit with different persistence lengths that was used for the simulation, and we picked the persistence length that gives the best quality of the fit. And you can see here the results again for, okay, so here we did it for different percentage of the stem loops formation for each one of those 3 structures that you see here. Without entering to the fine details, assuming that 50% stem loops is the more reasonable percentage of stem loops that will be formed, we get a persistent length of 2.8 plus minus 0.7. It's a little bit larger than what we had before, but still very close to that. And that's what we believe is the persistence length of single-stranded DNA. As a conclusion, what I showed here is the measurement of the single-stranded DNA persistence length by using DNA origami structures. Such single-stranded DNA may affect the chromatin condensation level, which is the long-term interest that I have in this field. And for future work that we already started, we are planning to insert such DNA origami structures into live cells for various studies, especially for trying to make them so that they'll take part in the gene expression procedure. Thank you very much for this, for the attention, and I would like to thank my students that took part in that, especially Efrat Roth, that's her main project. Also Hadass that is now starting to work on that. A lot of collaboration with scientists at Bar Ilan University, here at the Technion, and collaborations all around the world, including with Hao Wang and Lili Li from the NCLST. Thank you very much.
Okay. So thank you, Professor Garini, for a very eye-opening talk. I have my own few questions and also a question from Eitan Lerner from the audience. First, I was wondering, you were only looking at the size of the structures. Is there any connection to the actual sequence, or let's say for the percent of a single nucleotide that would influence these differences?
Okay, so on the extreme case, if, for example, a lot of the DNA sequence would be made from only one single nucleotide or only two of them, definitely there is an effect. It is well known. There is even a different bonding length to each one of the nucleic acid with each other. But the point is that if we look at the typical sequence of the DNA, it's pretty much mixed. So for such a mixed structure, I don't believe that the sequence has a lot of effect on the overall conformation, as long as you take something of the order of 500 nucleotides or more.
Okay, so thank you for that. And I'd like to present the question of Eitan Lerner. He was asking if you took in account the height differences between the ends, or did you rely only on 2D to 3D simulations?
Can you repeat the question? If we took into account
the height differences. Did you take into account the height differences between the ends, or did you only rely on the 2D to 3D simulations? That's the question.
Well, in the first structure, we only used the end-to-end distance, the first part of the work. In the second one that I showed, when we looked at the ring chromosome, there is no end point. And for these structures, we fitted the persistence lengths only based on the simulation. And the simulation is made by simulating the ring polymer in 3 dimensions, and then measuring the projection of it on the 2 dimensions.
Itamar wants to ask.
Yeah.
Please.
Yuval, very nice. As somebody that works on DNA nanotechnology, including origami structures, I have, first of all, I think that the use of AFM to address the question of persistent lengths is very interesting, but it has some limitations. Like, first of all, the yield of the correct structures on which you decide to focus the measurement, including the tip sizes that can vary and influence the results. So my question is, first of all, regarding the use of AFM, do you really see some limitations? And I would like to suggest you some nice complementary experiment. You are using origami. You can place very precisely a fluorophore, a donor and acceptor pair at the ends of where you connect the origami structures. Now, under these conditions, you can get from a fret measurement in solution, not on the surface, the suggested persistent length. Did you consider something like this?
Let me say, when we designed the project, we looked at all kinds of possible different experiments. Fret, by the way, was already used. I'm sure that you are aware of the works by T.J. Ha. He used Fret actually in one of the experiments in order to estimate the persistence length. The point is that with Fret, you are limited to relatively short distances. And when you do that, for example, in our case, in some cases, the end-to-end point was 20 nanometers, which is not exactly at the efficient Fret level, definitely not for the ring chromosomes. So we are aware of that method. Nevertheless, we decided to use the AFM. In terms of the limitations of the AFM, I would mainly point to the fact not of the problems that are associated with the tip dimensions and the convolution that you get when you do the measurement, of course, with the kind of point spread function of the tip. I would say the main uncertainty may arise from the question whether the polymer is really a two-dimensional projection of the 3D structure, and in that sense, what you suggested can definitely solve that, or does it allow the polymer, after adhering to the surface, to readjust itself? And if it does that, you should use a different kind of equation. We did not enter so much to that question. That was studied a few years back by Bustamante in very clear detail, and we just, let's say, use the conclusion that what the AFM is measuring is the 2D projection of the 3D structure. There's definitely some uncertainty, and it adds, I would say, variability to what we measure.
Thank you.
Thank you very much. I think we need, on the concern of time, we need to move on. So
thank you very much.
Thank you very much. Goodbye.
I think that we are moving now to Professor Tang and the Chinese presentations.
Yes, thank you. Maybe it's my turn to introduce Professor Wang Hao, our next speaker. Professor Wang Hao obtained his bachelor and PhD degree from Department of Chemistry at Nankai University in 2000 and 2005. Afterwards, he moved to University Würzburg in Germany as an Alexander Humboldt Fellow from 2005 to 2007. Then he was a postdoc researcher in UCLA from 2007 to 2010. Since 2011, he joined our center as a professor, joined our center, National Center for Non-Science and Technology. He was awarded numerous awards and have published 80 papers, 3 book chapters, and hold 8 patents. His research currently includes 3 parts. The first is develop supermolecular assemblies in cytological condition. The second is study their bio-effect and further regulate their biological behavior. And the third is explore supermolecular assemblies based imaging probes and drug delivery system. Yeah, we believe this kind of combination with the molecular assembly strategy and modern in vivo molecular diagnostics, therapeutics, and in vivo molecular diagnostics represents an important paradigm shift from drug delivery and clinical patient management applications. Yeah, I think Professor Wang works in this area, and not only in the fundamental research, also try to really transfer this technology to the real application. Professor Wang, please.
Okay, thank you, Professor way. So I'm gonna share my screening. Okay, here. Okay. Thank you very much for the Israel Academy of Science and Professor Wilner and also Gitaki. It's my great honor to share the recent program and the work from our group. Today, my topic will focus on the in vivo self-assembled peptide for bioimaging and drug delivery. As we mentioned, as we noticed, for the nanodrugs developed for a long time, I think the first nanodrug was approved based on the liposome, and the most famous one should be this one. We call it liposome, doxyl encapsulated liposome, and the trademark we call it doxyl, and which was approved by the FDA. Afterwards, more and more drug was approved by the FDA, and all this drug depends on not only for the drug molecule, but also some of cases related to the formulation. And this formulation, most of the formulations comes from the biomaterials and also self-assembled system. And what's the challenges for this drug delivery system? I think because of, as we know, for the drug delivery system, for the living system is quite complicated. For example, when the matter go to the blood, it will go to the tissue, cell, and even the organelle. And there have the multiple physiological barrier during the transportation, the whole system. So if we think about the nano system, because they have the small size and the controlled surface and interface and the quantum size effect, somehow it will show the better delivery system. That's why the nano carrier shows very promising in this field. But the problem is still right now, the delivery efficiency is quite low. If based on the literature report, people believe for the systemic administration, the delivery efficiency is below 1%. Of course, this number is still arguable, but anyway, we still need a lot of efforts to improve this delivery efficiency. So what we're gonna do for this system? Actually, from 10 years ago, and for our group, and we developed a new concept we call in vivo self-assembling, which means we can synthesize small molecule, then we can just self-assemble based on these building blocks in the physiological environment, such as cell, tissue, and even the living organism, to achieve the target accumulation and the retention effect in the lesion site. So why are we gonna do that? Because for the small molecule, normally this molecule have the smaller size, let's say less than 10 nanometers. For this small molecule, it's more easier to overcome the biological barrier to go to the lesion site. And when the bioactive molecule go to the lesion site, and because of this molecule can be triggered by the microenvironment factor, like the pH value, enzyme, and so on, it will self-assemble in this lesion site to form the defined and nanostructures. And due to these nanostructures have the bigger size and volume effect, this biomolecule can return in this area longer and show the better biological efficacy. Okay, this is a whole picture for our concept. And normally for this molecule design, and we have the modular design. For example, we have for this one, for this arrow, and means it's a target module. And for this module, we can target different kind of receptor and so on. And behind that, we have the stimuli responsive module. It can sensitize the different environment in the, for example, in the tumor microenvironment. And for the moreover, and we have the assembly module, and then we can trigger, once this molecule was triggered, then this stuff can self-assemble. And of course, here is the function module, can be the probe and even the drug. One small molecule goes through from the blood vessel, and it's just a passive transport to the tumor target. And then this molecule will target to the specific receptor based on the targeting molecule we designed. Then because this molecule, in the tumor microenvironment, they have a very special enzyme or pH. This molecule, for example, can be cleaved from here and form this residue. And this residue will simultaneously self-assembled into these nanostructures and form their function. For example, for this red dot, if it's the image probe, and it will go show the better image performance, right? And for the drug, we'll give the high concentrated drug here. Then after this structure finish their job, just disassembly, then to form the monomer will be clearanced from the body. This is the whole journey of the molecule from the blood to the end of the body. So today I'm gonna share the two stories along this concept. The first one, we're gonna talk about self-assembly inside of the cells. And we call it a tumor selective cascade activatable self-dictated system, TCAS system. The second one, we're gonna talk about self-assembling on the cell membrane. We call it addressable peptide assembling on the cancer cell membrane. So this is the first story, we call it the TCAS system. For TCAS, and this molecule, actually we have the 4 modules. And first one is recognition motif. Second one is enzyme degradable linker. And this is a self-assembled sequence here. And it will form the beta sheet structures based on the hydrogen bonding interactions. And later on, and this one we can just install the different function molecule, whatever. This is the NIR dye, right? Is we could Sci5, and all we can add the chemo drug. In this case, we use a DOX. And when the molecule get inside of the cell, and the first molecule will recognize this enzyme we call the XIAP. Then the XIAP will the caspase 3 and 7 will be activated, and the molecule will be tailored. The residue will self-assemble into this fibril structure and return in the cancer cell for a longer time. So this is a molecule design we perform. Here is the target part. It's a cell. It's a responsive. This self-assemble. Here is R. Is our function part like this. It's the imaging or the drug. And when the residue molecule will self-assemble into this very ordered fibril structure, and with a defined distance, it's quite a nice beta sheet structures. And for the molecule design, actually, when we do this full-length molecule, this molecule was self-folded into this sandwich structure. In the middle, the yellow part actually is hydrophobic and it's a very sticky part. In the beginning, this molecule was just the sticky part was encapsulated inside of this big molecule, and it cannot goes bigger and aggregate. And when the molecule was cleaved from here, the sticky part was exposed and initiated this aggregate. And based on our simulation, and we determine one critical parameter, we call it SASHA area. SASHA means the solvent accessible surface area. When this area reduced by at least 40%, this is a very critical number for the molecule design. So when we add the enzyme to this molecule, and we can see the molecule will self-assemble this into this file, very defined fibril structure, and with this and the layer distance are roughly 10 astro, and for the single strand distance about 4.7 astro. It's quite clear. And more importantly, for this concentration dependent study, and we realize this aggregate can be happen at very low concentration, even below 1 micromole. So that means this molecule very sensitive to this aggregate. And also we found this fibril structure inside of a cell. This is a cell slice, right? And inside of a cell, we found this fibril structure. And before we feed the material, we just labeled material with iodine. Because this iodine, actually the abundance of the iodine in this cell is quite lower. And after we labeled this iodine to our biomolecule, and from EDS, we can see very clear information from iodine, which means this fibril structure actually comes from our exogenous material. And also we also use other technique to stain and form this beta sheet structure. And based on this strategy, we can see actually this strategy is quite this is a uniform strategy to target different kind of tumor cells. And in this case, we labeled our molecule with different dye. One is a red dye, sorry. The other is a green dye, and both of the case, and we test the lung cancer, renal cancer, plant cancer, and they do show very positive signal, but for the normal cell, we didn't see a very strong signal. That means this strategy is quite good for the tumor target. And also for the mice study, and we also observe very good accumulation in the tumor. And this you can see for 5 days. This the top line is our positive molecule. Even at 5 days, right? You can still see in the tumor side it show very good signal. But for other control molecule, just degraded or clearance from the body. And in our case, the delivery efficiency is higher to the 10 to 15% based on the mice model after 48 hours. And also another advantages for this system is deeper tumor penetration compared to the bigger nanoparticles. In our case, the penetration distance around 60 micrometer. And also, it shows the lower systemic toxicity because of the non-self-assembled small molecule peptide will be easily clearanced from the kidney. You can see for this case, the liver retention uptake and 40 hours administration is less than 10%. So it's quite safe. And also we use this molecule to do some isolated human bladder tissue, and we perfusion our molecule to this bladder. And after 30 minutes, we do the NIR fluorescence imaging, right? And we can see this tumor inside of the bladder, and we can see very clear image from this tumor. And that means this technology potentially can be used for the image-guided surgery in the future. And this work we still collaborate with the PI from hospital, and project is still going on. So the second story is about the self-assembled on the cell membrane. So why are we gonna do that? I just want to show one slide, okay? Actually, this is a guideline from the European Association from the Urology. For one of the carcinoma, we call the renal cell carcinoma. And for this cancer, actually, this is a chemo-resistant tumor, right? That means for this tumor, any kind of chemo drug gonna be useless. Why is that? Because for this RCC tumor, the cell membrane is quite intact. the drug cannot get inside of the cells. So that's why we think, can we just self-assemble our material on the surface of the cell and see if we can sensitize this cell, make the cell from inert to sensitive to the chemo drug. So this is a molecule design and strategy. And we have this molecule, we call this target molecule with the DBCO. And first of all, this target peptide will selectively recognize this target, what we call the carbonic anhydrase 9, CAIX9. Then we add the second part. When these two guys, this AZI and DBCO click reaction to form the full-length molecule, they will simultaneously self-assembled into these nanofibers because of its extended hydrophobic interactions. And once it forms these nanostructures, the cell membrane will perturbation becomes loosely. That means the chemo drug can go inside of the tumor cell and kill the cells. This is the mechanism of this strategy. Okay, this molecule we got, we call one is P1 DBCO, and the other one is P2, we call the AZI, because this guy can click reaction very easily in the tumor cell, outside the tumor cell, right? So when this guy to meet each other, and we can see it form very good beta sheet structures. And also from the IR spectrum, we can see it also form the parallel beta sheet structure. It's quite clear to show this data results. And also for these two guys to meet each other, we can also say this is a UV experiment, because when these two guys meet each other, the solution will become turbid because it formed the aggregate, right? And if you see this curve and less than 10 minutes, a couple of minutes, the aggregate gonna be happen very quick system, right? So very efficient. And also we use the AFM to monitor the morphology of this molecule. And the 5 minutes, actually we see the very small dot. The size of the dot is roughly 65, 85 nanometers, somehow like that. But with time goes by and this aggregate grows bigger, like this one, the width is roughly 74 and length are roughly 200 nanometers, goes bigger. So, and also we feed our molecule with the cell, and we can see this is our positive molecule, right? And from 15 minutes to 4 hours, actually the molecule preferentially accumulate on the surface of the cancer cell. But for other molecule, have only a target one. It will just first go to the membrane, then go side of the cells and so on, and quickly degrade inside of a cell. And also we verify this structure by use of this giant plasma membrane vesicle. And we generate this vesicle from the cell solution. And because we treat the cell will generate this kind of giant vesicle, and we use the vesicle as a model. Here, actually, this is the vesicle generated from the cell. And then we just treated this vesicle with our material, as well as this physically labeled protein, we call it BSA. If this membrane is very contact without damage, and this protein cannot go inside of the vesicle. You can see there's no signal, a green signal at all. But once we have our material, because of membrane was perturbed, the green signal will go inside of the vesicle. That means our material can perturb this cell membrane. And also we use one probe like the NBD to monitor actually this aggregate can be happened on the surface of this cancer cell. you can see this strong NBD signal, which means further validate the formation of this aggregate on the surface of the cancer cell. Finally, we just use this our material with the conventional use of DOX. This DOX is kind of a chemo drug, quite normally used. And we see if we just use the pure DOX and in the cell nuclear, we barely see the drug signal, right? The signal is quite weak, but we use both of our materials and the DOX, we can see the DOX signal in the nuclear is much, much stronger. In that case, which means that our molecule can help the chemo drug get inside of the tumor cell. And in this case, we also measure the IC50 cell viability of this combination. And we see the IC50 of the DOX was reduced to 3.5-fold compared to the pure DOX. And also we just inject our molecule into the mice from the T1. And also we can see for up to the 8 hours here, you still can see very strong signal from our material. That means that our molecule can long-term return in the tumor site and give the better accumulation effect. Yeah, here is the Sn ratio of this material. Finally, we use this strategy to treat the tumor-bearing mice. This is a mice. Actually, our protocol is like this. First of all, we inject the first molecule we call P1 BBCL from T1. After half an hour, we inject the mixture of the second molecule and the DOX chemo drug. So based on this protocol, after 3 injections, and if you look at the purple curve, right, compared to the free drug or control and so on, and we can see the signal, the signal is much lower, and we can well control the growth of the tumor cell. Yeah. So finally, I want to thank all of my group members and also funding agency for their support to our work. Thank you very much.
Thank you very much, Professor Wang. That was a very interesting talk. I would love to hear some questions from the audience, but maybe I will start with your first part of the first half of your lecture when you were talking about the compounds that, you're talking about the compounds that penetrate the cells and then they actually form your beta sheets. I was wondering, how do these compounds actually get into the cells? Because they're very large compounds and they should be very, loaded with hydrogen bonding. So that really shouldn't, on paper, that should not really work.
Okay, so that's very good question. So the quick answer is that the cell uptake mechanism is not quite clear though yet. So, but actually the result is very clear. And we can see this molecule, big molecule can get inside of the cell. And right now we believe, because the size of this molecule is roughly 3,000 Dalton, right? So it's a big molecule. And we believe because of this, for this molecule, because they somehow have the positive charge, and also for this molecule, for example, we have this IR dye, and based on the literature for this IR dye, maybe they have a very special receptor-mediated pathway to get inside of the cell. But all this is a hypothesis. So far, we still work on this system and to finally verify the mechanism, the cell uptake mechanism. But for the results, we actually 3 or 4 students repeat this experiment, but the result is positive, but the mechanism is still going on.
So I will ask on you and Professor Wee for 1 second, but I would still want to continue here with this. My field is proteases, and my feeling is that these big molecules get cleaved not inside the molecule, not inside the cell because of caspase 3, but outside of the cells because of other proteases that are highly overexpressed in the cancerous environment. So the sequences could actually hint on that. And there's also ways to check. We can talk about this later, but my gut feeling is this compound gets actually cleaved before it even gets into the cell, and then it gets into the cells, and then it could aggregate.
Yeah, I cannot rule out this possibility. The molecule get cleaved outside of the cell in the tumor microenvironment and so on, right? Yeah, actually for this enzyme sensitive linker, actually we tried a different linker. Some of the linker is tumor sensitive, but some of them is not. So far, I'm not sure the major point for this major site for this cleave is outside, but yeah, but we still wait for the more data to support this hypothesis. Yeah, but that's very good suggestion.
I mean, we have tools to look at that, but thank you very much. And Professor Wee, do you want to ask your question?
Yeah, just a very short question for the self-assembly. Actually, yeah, in the most rare cases, you get the nanofibers or fibril structures. I'm not quite sure if the morphology change is any effect on the diagnostics or the biological functions with these assemblies.
Okay, that's a good question. So for the morphology control, so right now we can form the nanoparticles and also the fibril and also the hydrogel, yeah, inside or outside of the cells. And for the nanoparticles and the nanofibers, actually we didn't see the dramatic effect on the cell feed or life or the viability, but definitely for the in situ formation of this hydrogel inside of the cell, right? And we can observe the cell just detach from the culture dish and died immediately. So that's very interesting phenomenon we observed for this from this different morphology of the self-sampled structures. Yeah.
Okay, thank you.
So another question from Eitan Lerner from the audience. He is asking if you're not worried about the nucleation of other aggregated proteins that will start aggregating because of this process. So maybe this aggregation will actually cause other proteins to aggregate that you don't really want to aggregate.
Yeah.
So this is the question.
So, okay, that's a good question. So we talk about the safety issue and side effect issue of this this aggregation. So actually in our living system, actually we have a lot of aggregation system, and day and night we just produce this undesired waste protein aggregation. Actually in our system, for example, we have the autophagy system or mechanism or some other mechanism to clearance all this stuff. But if we can control this self-aggregation in the desired site, for example, in the tumor, might be the positive contribution will be more better to kill the tumor cell compared to some other area. Probably they have some aggregation, even induce some other protein aggregation and immunogenicity and so on. But depends on dosage we use and the amount for the different organs. So far, we didn't find so much toxicity issue and immunogenicity. Yeah, it really depends on how to control and the dosage we used. Yeah.
Okay, and his response is this aggregation after the depletion from the tumor, this actually might this might circulate for longer periods of time then. So this aggregate of other proteins.
Yeah, but I think it can also be disassembled, because we also do this long-term monitor of this aggregation, and it's not like the stone, like the solid stone and just circulation in the body forever. But gradually it will disassembled finally. So.
Okay, so thank you very much for that talk, and now it's our time to move on. So,
okay, thank you, Galia. Thank you, Zhixiang.
So I'm trying to introduce the second Israeli speaker, Professor Ariel Ismach. Professor Ismach got his Bachelor of Engineering from the University of Ben Gurion in the Negev. And then after completing his bachelor, he moved to the Weizmann Institute, where he got his master's and PhD under Ernesto Yusilevich. At the end of his PhD, he went to Berkeley for a 2-year postdoctoral. Later on, he moved to the University of Texas in Austin, continuing his postdoc. And in 2014, he returned to Israel. He got a position in the Material Science and Engineering Department at Tel Aviv University, where his laboratory is currently dedicated to study the growth and properties of two-dimensional atomic crystals. His group is working to address basic scientific questions regarding the formation and the structure property relation of 2D materials, as well as developing new methodologies to engineer layered materials and their heterostructures for application in catalysis, energy storage, and optoelectronics. Professor Ismach, please, the stage is yours. 25 minutes.
Thank you very much for the kind introduction, and thank you very much to the organizers for inviting me to this workshop. And let me just a second. So, as Professor Gyration introduced me, my field of study is kind of different from the two speakers, the previous two speakers, so I hope that it will be okay for the audience. So I will be talking about epitaxial growth, epitaxial organization of atomically thin semiconductors, which are basically layered compounds at the single or few layer level. These compounds, I'm not sure how many of you are familiar with these unique compounds, are very unique in the sense that they are highly anisotropic. So they have covalent bonding or ionic bonding inside the layer, but in between the layers, they have relatively weak van der Waals interaction, and therefore we can basically isolate single or few layers of these materials. And also because of this anisotropy, we can grow them at the single layer level without the need of unique equipment in ultra high vacuum and so on, such by, as I will show you in a minute, very standard and simple equipment. So in this slide, you can see that this family of layered compounds is huge. There are many, many types. This is just a partial list. But you can see that they cover basically the whole spectrum of the energies, the band gap energies needed for a huge amount of applications in catalysis, energy, semiconductors, electronics, optoelectronics, and so on. And even more than that, you can see that in some materials, the band gap spans in a wide range of energies. And this is because these layered compounds, the physical and chemical properties of these layered compounds are highly related to their structure, namely the number of layers, the registry between the layers, the chemical composition, and so on. And therefore, for us, material scientists, these materials offers us a huge playground to grow and study in order to synthesize new functional materials and heterostructures. So yes, our lab focuses on growth mechanism studies, and basically we are very much interested in studying the processing, the synthesis with the structure, chemical composition, number of layers, registry, and so on, and correlate all this with the physical and chemical properties. So, in this talk, I will describe briefly how we can grow these atomic layers at the single or few layer level. I will give some examples. And then I will move on to one of the key questions here, which is we know that these materials are very interesting. They have unique physical and chemical properties, but one of the bottlenecks in order to try and do further studies or implement such materials into novel and existing technologies would be the ability to grow them with the desired properties, which usually refers to the crystallinity, number of layers, and so on. And within this topic, I will describe our attempts to do so by what we call the seeded growth approach, a growth edge approach, and finally, which is the ultimate, the optimal thing to do, in my opinion, would be the epitaxial growth. So when we talk about the synthesis of 2D materials, there are, of course, many methodologies to do so. I will cover only basically the vapor phase methodologies, which are considered to be very promising to the large scale synthesis of these materials with the desired properties that I described earlier. Now, if we dig into the, and usually these methodologies rely on what we call the chemical vapor deposition. Basically, you have a reactor, you flow some gases, which are the precursors, and you heat up the system, and by thermal decomposition or sometimes there are catalytic decompositions, and you have a chemical reactions which leads to the formation of the different phases, which is the basic of chemical vapor deposition. Now, when we deal with the chemical vapor deposition of these 2D materials, the vast majority of the works, because of issues that are not related to the output, but are related to the easiness and the ability to handle these precursors, the vast majority of these works, including some of our works, actually we work with precursors that are not suited for chemical vapor deposition. We use them because they are easy to use. But as a result, we get layers that are highly non-uniform in terms of organization. Not all of them are planarly organized on top of the substrate. They can grow as vertical layers. So we have some regions with vertical layers, multi-layer growth, and then we may have some nice regions with one layer. And I will explain in a minute why in some materials, sorry, in some materials it's so important to be able to grow at the single layer level. So yes, so these methodologies usually leads to very highly non-uniform films that are basically not desired. In principle, you can improve them as we did and many others did, but these precursors are not the best. Now,
Ariel, I'm sorry to interrupt. We see only the first slide for some reason.
Oh.
I'm sorry to interrupt, but we don't see the slides.
Now you see the slides?
For the first time, we see a different slide than the first one.
So I apologize about that. So I showed you this diagram with the different layered compounds. Thank you very much for letting me know. And do you see the slides now running?
Yes.
So this is basically the simple setup for chemical vapor deposition of TMDs or 2D materials in general that many groups around the world uses. And I will explain why these methodologies are not the best to achieve the desired material. As you can see here, I was explaining later, you weren't seeing that you can get vertical flakes or multi-layer areas or very non-homogeneous films. And basically we can, by the same methodologies, we can show that by tuning some parameters, we can end up with totally different materials, from inorganic fullerenes to core shell metal oxide and metal disulfides, and to one layer growth domains, as you can see in the final, in the bottom of the slide. So when we talk about the one layer, especially in this family of 2D materials, the transition metal dichalcogenate, which is basically an array of transition metals which is sandwiched with two chalcogens, the importance of having this as a single layer, you can see that from this first paper in 2010, where they proved that these materials, as we knew from tens of years, these materials are what is called an indirect band gap material, semiconductor, which means that in order to have a transition from the excited state to the valence band, you need to have a momentum change and also the energy change. So then the probabilities to have photoluminescence are very low, as you can see here in green. So this is the photoluminescence measurement in a two-layer MOS2 in this case. But when you go down to a single layer, the PL emission increases dramatically, because it turns out that from the bulk to the one layer, there is a transition from the indirect to a direct band gap semiconductor. So here you can see an example in which you have a single crystal domains, so in this case MOS2, and again, every domain like that is a 3-atom thick material, the transition metal at the center and two chalcogen atoms bottom and top. Now, one of our methodologies to try and improve this the growth of these materials, and also to study the mechanism, was taken from many other fields. For example, in a 3D crystal growth or even colloidal growth, and is what we call the seeded growth approach, where you start the nucleation from well-defined seeds. And by doing that, in many fields, you can, in principle, control the crystal orientation, the shape, the size. In colloidal synthesis, you can control the size distribution, for example, the chemical composition, and so on. This is the basics, for example, of our silicon technology as we know it today.
Sorry, Professor Isbach, we see only the first slide of your presentation.
Wow. So I don't know what's the problem, so I guess that I will continue like that. Do you see that? I will not continue in a presentation mode. Do you see now the slide of the crystal growth from seeds?
We see only the CVD derived transition metal.
It's the first time that happens to me.
Maybe try to go to.
I will stop the share. I will reshare again.
Maybe try to share it again. Thank you.
Sorry for that again. So I was talking to myself. Okay. Okay, now you see the right slide. I will continue on not in the presentation mode. I apologize about that. This is the first time that I have this problem. So when we implemented this seeded growth approach using a very simple photolithographic methodology in which we pattern basically islands of different metals. Here you can see islands of gold. And then we do the chemical vapor deposition. And then you can see very nicely that as long as the surface diffusion of the different precursors is larger than the seed distance, the seed pitch, basically, so then you have a preferential nucleation at the seed location, and therefore you can continue the growth from this seed. And this is the main idea. So then by doing that, you can in principle control the crystallinity of your film, because you can control the domain size of your film once you continue the growth and you merge the domains. Here you can see an example, for example, in which you have in the same sample, you have seeds that are separated by more than 50 microns, and seeds that are separated by 10 microns or 20 microns. And you can see the merging occurs only, of course, when the seeds are denser, because there you have higher nucleation density, which are basically the seed density. So this is nice, of course, and a few other reports were showing the same. But in this case, you need to be careful because some of the metal seeds might affect both the crystallinity and also the chemical composition of the, in this case, TMDs, MOS2 or tungsten disulfide. And as you can see here, for example, from photoluminescence maps, so we can do mapping of PL of these domains, and we can show that there is a strong correlation between the presence of gold. This is by TOF-SIM, so this is a chemical mapping, and this is a photoluminescence mapping. We can correlate between the position on the concentration of the gold along the domain and the PL, both intensity and shift. And we are not going to talk about that in this talk, but the point here is that if you're not aware of that, the metal may diffuse into the domain and affect greatly the physical properties, in this case, the optical emission of these domains. Now, as I was mentioning before, the standard CVD approach that used basically precursors that were located inside the growth system, inside the reactor, these are not the best suitable precursors to work in the chemical vapor deposition schemes. The best thing will be to have precursors which are located outside the reactor. So this must be, of course, volatile precursors. So these are a totally different type of precursors that if you manage to put them outside the precursor, so then you can look at them as, a Lego, right? So every precursor a different color. So in principle, during the growth, you should be able to switch between the precursors, because now we totally control the delivery of each precursor to the growth area. And therefore, we should be able to in situ grow very complex heterostructures, which is also one of the goals of my group, to study the growth of such structures. Now, of course, there are a lot of technical issues with that. Every precursor have a different kinetics and so on. But as a proof of concept, you can see that here, by switching between molybdenum and tungsten precursors, we can basically make, here you can see a 3-atom film, okay, all over the substrate. But in some areas, you can see that there are domains, and we can prove that by Raman, PL, and of course, chemical mapping. So we have MOS2 domains, which are surrounded by tungsten disulfide film, all of them 3-atom thick. So this is a nice proof of concept. But what we want to talk about today is about the intrinsic growth of this material. So I was mentioning that one of our goals is to improve the quality, to understand the growth, and then to improve the quality so we can push further these materials for both basic studies, but also different applications. Now, one of the methodologies that we may use and consider, and there are a few groups around the world considering that, is a special case of chemical vapor deposition, which is with the use of metal organic complexes as precursors. This is well known in the industry. So the problem with these precursors is that they thermally decompose at relatively low temperatures, and therefore the density of nucleation in this case is huge. And we end up, as you can see here in these three images, we end up with basically nanocrystalline films. So here this film was transferred to a different substrate. So the purple color is the color of the target substrate, which is a 300 nanometer silica on top of a silicon. And but this is TEM and this is electron diffraction. So then we adopted a methodology in which we can, again, because we move everything out of the reactor, we can introduce different gases and different precursors, and we can start trying to introduce vapors that will clean the layer during the growth. As you can see here, the Raman, so in green, you can see the Raman the Raman spectra from these layers. They contain a significant amount of amorphous carbon, and this is expected from metal organic precursors if you don't control that well. But once you introduce very low amounts of water vapors or oxygen, you can significantly enhance the Raman signal, which is an indication of improving the crystallinity of the films. You can increase the domain size, and also you can effectively re-evaporate the amorphous carbon present in the amorphous carbon impurities by just making CO and CO2 vapors. So you will re-evaporate that to the reactor, de facto cleaning your film from carbon impurities. And then we can show that if we move on and we implement a sequential growth in which we grow and we etch, by pulses, we can increase dramatically the domain size, which is again an indication of crystallinity. Now you can see the atomic resolution TEM images, and also the domain size in the range of tens of microns, and also, of course, PL and many other techniques. And the idea behind that is that, okay, I will explain the idea here. Okay, so that was an animation anyway. So in a standard growth, you have the nucleation of the TMDs in blue, in this case, tungsten disulfide in blue, and then you have some carbon impurities along the way. These carbon impurities act as further nucleation points, and also encapsulated in the films, so therefore lowering the crystallinity and the quality of the film. So as the growth continues, you end up basically with a continuous film with very small domains and with carbon impurities, as I showed you in the beginning. Now, when you implement this growth and etch pulses, basically we grow the layer, we start the same growth as I showed you before, and then we etch with, we stop the growth and we etch with very small amounts of, in this case, water vapors. So we evaporate not only the carbon impurities, but also we evaporate small and defective domains that were grown in the beginning. And therefore, we not only clean the surface from carbon impurities, but we basically allow for the growth and allow for the higher quality domains to continue the growth. And we end up with highly crystalline domains and highly crystalline films. So this is the basic idea of the growth and etch methodology. And again, you can see here, for example, images of different sequences. We're not going to get into details, but different sequences showing the increase in the domain size, so the brighter domain, the brighter triangles or the isolated domains. And eventually we can grow single layer wafers using this methodology. And we can compare the optical properties of these materials by stationary PL, by temperature-dependent photoluminescence, and also time-resolved photoluminescence at the Weizmann with Professor Danaron. And we can show that these material properties are comparable to the best materials in the market, which are the isolated flakes from 3D crystals, which is very encouraging. Now, I would like to move to the final idea, which is, I think this is the optimal case in order to have to improve the quality of the film and to have a higher quality available. And it is, of course, the epitaxial growth of these TMDs. Now, you need to understand that in epitaxial growth, in a classical, let's call it that way, the classical epitaxial growth means that there is a clear commensurism between the growth substrate and the film. And this commensurism basically implies that there should be a mismatch difference between the lattice's parameters of the substrate and of the film. If that mismatch is too large, so epitaxial growth is not possible. However, in van der Waals layers in 2D materials, because there is no covalent bonding, just van der Waals, that means that you can grow in principle van der Waals epitaxial growth, even though there is no commensurism. And therefore, you can grow layers that the mismatch with the substrate is much larger than the allowed in the commensurism epitaxial growth. And this was, the van der Waals epitaxy term was coined by Koma about 30 years ago. And since then, of course, many 2D materials on 2D materials or 2D materials on 3D systems were shown to be growing. We also grow epitaxially different materials. Here you can see an example of a different layered compound. In this case, indium selenide, indium-2 Sc3, which every layer has 5 atoms, a quantuple. And usually this material is grown on mica, and it grows epitaxially. Mica is also a 2D material, very complex 2D material, layered compound. But when you grow that on mica, the problem is that since the mica is very complex, it's very hard to study the growth mechanism and the epitaxial relation. And also, if you want to study the electrical properties or other properties, and you need to transfer these layers from the mica to a target substrate, you end up with sometimes with chunks of the mica, because also the mica is a layered material that can be exfoliated. And therefore, we implemented an epitaxial growth on TMDs, the same material that I showed you before. We can show that this indium selenide actually grows very nicely epitaxially on this MOS2. We can show the growth evolution by AFM Raman, and so on. And we can basically prove that this is epitaxial growth. Here you can see basically this image is a collage of the data from 3 different transmission TEMs. So here you can see electron diffraction from different points. So here you can see a single domain of MOS2, which is coated with few layers of indium selenide. And here, the diffraction peaks, all of them confirms the epitaxial relation. And then, of course, you can have in-plane atomic resolution showing the superstructure of these 2 dimensional layers. And finally, the cross-section shows very clearly the single layer MOS2 and the indium multi-layer indium selenide, in this case, really thick, on top of the MOS2. So by all this, we can prove that it is indeed growing epitaxially. So therefore, the domain size of the indium selenide is dictated by the size of the, in this case, the MOS2. How much time do I have?
Well,
okay. 3 more minutes. Okay. So, and with regards to the growth of TMDs by such van der Waals epitaxy, it was shown by a few reports that by the same methodologies or similar methodologies that I showed you before, this TMDs can grow epitaxially on, for example, on C-plane sapphire. And from our experience, we noticed that you can get epitaxial growth or you cannot get epitaxial growth. We invested a lot of efforts in trying to understand what are the key parameters here, and we couldn't find the answer for that. And finally, after several months of hard work, my PhD found out that when you do prior to the growth, when you do a in situ seeding, metal seeding approach, so you basically expose the substrate only to the metal precursor, so by that creating nanoparticles of this metal on top of the sapphire. By doing that, we increased the chances to get epitaxial growth from like 10% to more than 95%. So that means that this seeding approach is significant for this epitaxial growth. You can see here some results. But the important thing is that if this is the case, if you need to have a metal seed which is epitaxially grown on top of the sapphire as prior to the growth of the TMD layer, that means that the van der Waals epitaxial growth mechanism that was explained so far, which is the arrangement of 2D materials on top of 2D materials, but without any commensurism, might not be accurate. So what we are doing these days is trying to further understand this mechanism, but also to ask ourselves how general is this mechanism. Is it general to other 2D materials of many different types, or only to this transition metal like alcohol? Okay, so I guess that I finished my time. I apologize again for the presentation in the beginning, the troubles. I would like to acknowledge my group, of course, so the guys that actually did the work, Asael, my PhD, and a few of the postdocs, Abinash and Pranan, and then collaborators here and in many other places, and of course funding. And thank you all for the invitation. Thank you the organization for the invitation, and thank you the audience for the patience. Thank you.
So thank you, Professor Itzmach, for a very interesting talk. I'm not very familiar with this field, and I directly am trying to connect this. Is there any relationship between your 2D growth and 3D growth? If the seeding is on a metal, like you just described in your last few slides, can you translate this into things that are in 3D in solution and not on a bed or on some kind of silicon matrix?
So the problem in growing these materials in solution, and there are some wet chemical approaches to grow these materials at low temperatures, like 150 or so. The problems with that are that in this case, the layered materials grow very defective, and they grow usually as flakes, vertical flakes. There is no control over the arrangement and also the crystallinity. Now, this is good for a lot of applications like catalysis and maybe water cleaning processes in which you need a high density of defects in order to react with many other species. But for other applications, chemical approaches, they don't work. You can grow multi-layer, and we can also show that multi-layer films by using basically the same methodologies. Once you understand what nucleates these films and how to continue the growth, so then you can implement that in order to grow multi-layer films. We usually are focused on the single or very few layers, which is the regime in which we can study the growth mechanism more efficiently.
Mm-hmm. Very interesting. Are there any more questions from the audience? So I think for lack of time, we're already a bit late on the schedule. I think we will move on. So thank you very much, Professor Isma. Professor Gedanken, you want to?
Oh, that's now we are moving to the Chinese presentation. So Professor Wai, will you take over the stage?
Yes, thank you, Professor. Yeah, and next I will introduce Professor Aiguo Wu from Ningbo Institute of Material Technology and Engineering, Chinese Academy of Sciences. Professor Wu graduated in Nanchang University in 1998 and received a PhD at the Changchun Institute of Applied Chemistry, Chinese Academy of Sciences in 2003. He has ever worked in University of Marburg in Germany and Caltech and Northwestern University in US from 2004 to 2009. He returned China and joined Ningbo Institute of Material and Technology, Material Technology and Engineering, Chinese Academy of Sciences as a professor. His research interests are mainly in the nanomaterial in biomedical imaging in cancer serologics. Professor Wu have awarded a lot of honors. For example, the National Science Fund for Distinguished Young Scholars in 2020. He have published more than 200 papers and was also served as a journal editors as a journal editor board members. So Professor Wu, please, we are looking for your presentation.
Okay, thank Professor Wu's nice introduction. And also, I'd also thank all the two countries organizers to give us these very nice opportunities to show our recent work. Today, I will show you the work is called MPY-based cancer diagnostics. This I go from the Ningbo Institute of Materials Technology and Engineering, Chinese Academy of Sciences. And I think before I introduce some work, I should tell all the people where I'm from. I'm from the Ningbo. It's a nearby Shanghai city. It's a hub city in China. And also, I think is the biggest one hub city for the freight ship around the world. And here is a hub city. So, and our institute was founded in 2004. And up to now, we have more than 1,200 staff members and 1,600 postgraduate students. And this institute was founded by Chinese academic sciences and the Zhejiang provincial government and the Ningbo municipal government. This the main campus view at the Ningbo main campus. And also we have 4 division institutes, including the New Materials Institute, New Advanced Manufacturing Institute, and the New Energy Institute. And the 4th institute called the Cixi Institute of Biomedical Engineering. It is located at the Cixi, and here is the Cixi campus. And this is around the Cixi campus, around the environment for the students and the study and life. And this is our group. Our group, we have over 70 members in the groups, and including the professors, full professors, associate professors, guest professors, senior engineering, postdoctors, and students. And our research interest focus on the nanomaterials and their applications in biomedicine, and they're including the tumor slurry material and the technique, and the MI probers, and also the nano contaminant detection probers. And today I want to show you is the neuropeptide tumor targeting prober. And also we get a lot of international collaboration, because today we are also showing for the peoples, the 2 countries to seek for the potential in the collaboration each others. And in 2015, we published a paper just about the neuropeptide use the nano systems. So why we choose this system to our work? Because the neuropeptide is a kind of peptide with the 36 amino acid peptide, and it was discovered in 1982. It's almost 40 years ago. And in the human body, there are 4 subtypes: Y1, Y2, Y4, and Y5 expressed in the human body. And the main location area is central and the peripheral nervous systems. And it's with the function around, for example, food intake, anicity, and cancer. And since the neuropeptide Y it was discovered in 1982, but the structures until the 2018, it was discovered by a Chinese group, leading group in an international collaboration group to get this one. Since then, there are a lot of people just want to know how to use these structures to get more pharmaceutical things to do some things. And according to the literature research, we found there are some kinds of the cancer related to the neuropeptide receptors. For example, breast cancer, adrenal-colonic tumors, glioblastoma tumors. And in 2010, a group from the Leipzig University in Germany, they published a paper, used the MPY, attach the PET, the PET probers to get the PET MPY imaging. So it is possible to use the neuropeptide receptor as a promising targeting site for the tumor imaging and the therapy, but particularly use the nanosystems. So our answer is maybe possible, because we know the nanotechnology, there are a lot of applications in the cancer based on the properties, and also related to the tumor microenvironment. Just now, Professor Hao Wang from the National Center in Nanoscience and Technology of China showed a very exciting data for us. And also, it can show because the tumor microenvironment related to the pH, GSH, hypoxia, enzyme, and so on. So in the last several years, we used the Y1 receptor ligands for the tumor imaging, for example, the ultrasound imaging, fluorescence imaging, magnetic resonance imaging, MRI, and the computed tomography, CT imaging. And also, we want to use the ligand to attach the therapeutic function group, for example, to get the chemotherapy, photodynamic therapy, phototherapy, and also to get the very high affinities. We are also designing the new ligands design for the use the Tianhe-2 National Superconducting Computer Center in Guangzhou to design some new ligands to get the very high affinity there. So we used the nano system, including the albumin nanoparticles, polymer micelle, ribosome, up conversion nanoparticles, metal organic framework. So let's go to the detail. The first one I want to show you is for the breast cancer Y1 receptors, because in the normal breast tissue, they are expressed the Y2 receptors. And here we get four sites mutation for the Y1 receptors at site 30, 31, 32, and 34. And then attach the albumin nanoparticles on the surface to get the targeting function there. So here is the scheme and how we use the albumin nanoparticles coating the doxorubicin inside, and on the surface we attach the PNBL, MPY to get the targeting and to get the therapeutic function there. And we found the cell uptaking of the PNBL, MPY, doxorubicin, the albumin nanoparticles in the human breast cancer, like for example, MCF7, we found the cellular uptake was increased about 65.8% by PNBL and MPY modification. Further, we found the cellular uptake difference was increased about 1.9 times compared to the normal cell. For example, MCF7 10A cell is a kind of the normal breast tissue cell. And the second case I want to show you is we attach the nanobubble to enhance the ultrasound enhancement to get this one. And here we prepare the tumor. The bubble is about 200 nanometer around them. And then we use the DSP PEG attached on the surface. And these materials from the Professor Jian Yang from the Pennsylvania State University. And then we form the nanobubbles, and to get the nanobubbles inject into the tumor area and then get the ultrasound enhancement there. And we can find it can in vitro can improve the in vitro targetability for the nanobubbles. And we found in ex vivo fluorescence imaging found that in the tumor area is the signal is better than the liver, kidney, lung, and spleen. And it means most of the nanobubbles goes to the tumor area. And for the intratumoral injection, we found the instituted contrast enhancement get very high the enhancement. And we found the contrast enhancement over 40%, 42% in the intravenous injection for the in vivo ultrasound enhancement imaging. And the third work I want to show you is we use the AP-MPY, and at this time we get the mutation for the MPY at site 6 and site 34 to get the higher affinity to Y1 receptors. And also we just want to avoid the anti-photocytosis and use the self-peptide to get this immune escaping for this one. So here it is showing the scheme for our research work, and we found this kind of the system is a kind of the pH protective drug release, and also it they formed the tetramer at a normal pH is 7.4, and also at the lower pH time, for example, at the 5, it diplomized at pH 5. And we found also this system can decrease the IC50 about 80%. And also, according to the MRI contrast, and also we found it can enhance the MRI signal, and also can reduce the accumulation in liver and the kidney. So usually the nanoparticles accumulation there, but after conjugated with the AP-MPY, the accumulation get reduced. And also we found the very good the chemotherapeutic function. For example, it reduced the tumor volume about 32%. And also the nanoparticles can let the mice get prolonged the survival time to 100 days. And for the control group, all mice are dead. The first work I want to show you is the Y1 ligand used for the glymer imaging and chemotherapy. This work we are collaborating with the Institute of Automation, CAS, in Beijing. And because the Y1 receptors with very high expression rate in glymer's, so here we design a DSPG-MPY and get the nanomaterials to get the self-assembling there, and also attach the iodide and the doxorubicin. And here time, for the iodide, we can get the imaging, and for the doxorubicin, we get the chemotherapeutic function there, and to get the purpose. And we found the U87MG tumor in bearing mice can get very good imaging and improve the in vivo targetability. We found 2 kinds of the MPY mutation. For example, the AP-MPY is better than the PMBO-MPY. And further, we found this prolonged the survival time and the rate. The AP-MPY is also better than the PMBO-MPY, and get very good high affinity there. And also we get the accumulation, for example, compared to the the free iodide and the nanoparticle iodide, and also the PMBO-MPY iodide, and also this AP-MPY that we can found the nanoparticles can go through the BBB and across the BBB, and also a lot of the nanoparticles can accumulation inside the brain tumor area. So why we can get the AP-MPY is better than the PMB-MPY, and we use the simulation. We found the binding energy is AP-MPY is better than the PMB-MPY, and also in the plasma, the stability also the AP-MPY is better than the PMB-MPY. And just now I showed you for the therapy or imaging. So can we also use this nanosystem go to the multimodality imaging and the therapeutic function? Here we want to show you a case. We design a nanoparticles with the CT enhancement, upconversion fluorescence enhancement, and also magnetic resonance image enhancement, and also the a component with the photodynamic therapeutic functions after the MPY targeting on the nanoparticle surface to get the triple modality imaging enhancement and also photodynamic therapeutic function there. So we found it definitely can enhance the MI, CT, and ultra upconversion fluorescence contrast there, and get a very good signal enhancement. And also we found it also improved the photodynamic efficiency about 12% for the tumor bared mice. And also the challenging for the breast cancer, especially a subtype of the breast cancer, triple negative breast cancer. So here also we use the MPY as a targeting molecule for there, because we found the AP-MPY with very positive high expression rate for the triple negative breast cancer. So here we use the nano system is based on the metal organic framework because it consists of the metal ions or clusters coordinated to the organic ligands to form 1, 2, or 3 dimension structures. And also, for example, different morphs, there can easy modification and with high loading capacities. And also it is biodegradable. And for this one, we can show, for example, this morph with pH sensitivity degradation and also with high circulation stabilities. So here we choose the 2 kinds of the morphs, for example, ZIF8 and ZIF19. And we found that ZIF19 with faster self-assembling process and also facile post-modification and with lower cell toxicity there and high survival rate. And we found it reduced the premature drug release at the physiological pH level. And also there is kind of the pH and ATP dual responsiveness triggers the fast drug release. And we found also the better tumor cell membrane and the mitochondrial capabilities decreased the IC50 value about 40%. So up to now, we didn't know why there is a kind of the cell membrane and particularly for the mitochondrial capabilities. So for the reason, we are also researching. And we found this nanosystem can reduce the tumor volume by over 30% and also increase the survival rate about the 80% there. And another case I want to show you is we want to use the Y1 receptor ligand to get for example, some imaging guided chemotherapy there. So here we resort to a system, magnesium ZIF19. So here we use the magnesium ions, exchange the zinc inside the ZIF19 to get the T1-MI signal. And also these can let the T1-MI guide the chemotherapy there. And because this system can reduce the drug release during the blood circulation and also can with the cell membrane and the mitochondrial dual targeting there. And we found the size is for the nanosystem here is 18 nanometers and with the negative zeta potential and with very high drug loading rate there. And we found use the Shanghai synchrotron facility. We can better the data found the tumor cell membrane and the mitochondrial targetability there with very good signal enhancement. And also, it is a kind of the pH protective drug release about 70% and also decreased the IC50 about 52%. And it is enhanced the MI contrast about 49%. And we found these nanoparticles can most accumulated in the tumor area. It found the increased the accumulation in tumor about 2.2 times compared to the control group. And we found this kind of the nanosystem can increase the blood concentration about 1.9 times and reduce the tumor volume 42%, increased the survival time 45%. And to get the very high affinity there, so we are also design the Y1 receptor modeling and to get the novel Y1 receptor ligand there. So we collaborate with our colleagues at NIMIT CAS and also with the Upasana University, Professor Danahama, and for their help to get the very high affinity there. For example, we get after select one of six ligand for example, Y1 receptor ligands, we found that they can cross the BBB and with the glymer dual-tactin. And also we find a kind of a very interesting synthesis, the L-type, the AP peptide, with compared to the D peptide, they are different. The D-type APT has 2.5 times more hydrogen bonders than the L-APT. And also we found the T-types Y1 ligand probers with dual tactin of BBB and glymer and the liposome escape. Further, we found the nanosystem with the taggabilities of the D-type of the APT nanoparticles was 3 times higher than the L-APT nanomaterials in vitro. And further, we do the animal model found the D-type the APT nanoparticles was 3.75 fold higher than the L-APT nanomaterials. And also, the taggability of the DL-APT nanoparticles is mainly induced by the Y1 receptor to cause these things. And the further work, we found the D-type APT nanomaterials was 2-fold higher than the L-APT nanomaterials. And we found this is very interesting, the novel Y1 receptor ligand and the glymer imaging and the SLAP there. And also, we found the survival rate of the D-type APT nanoparticles was about 80% within 30 days observation for the mice. To sum our work today, I want to show you is first, we elucidated the tactin mechanism of the MPY nanosystems and for the Y1 receptor internalization and the physical chemical properties of nanosystem, for example, the size, the potential components can do, and so on. And the second one is we developed the pH protective release strategy of the Y1 receptor ligands, reduce the premature drug release from the nanosystem and increase their accumulation in tumor areas. And further, we designed the novel Y1 receptor ligands based nanosystem with high specificity and sensitivity for the Y1 receptor overexpressed tumor imaging and SLAPI. So what is the next expectation for this area? We think the next work we should do is ensure the self-assembling and the modulation of the neuropeptide nanosystem for the tumors or the central neural systems disease, for example, the Alzheimer's and so on, and Parkinson's there. And also, I also thank my collaborations around the world and the funding agents from the China area, and also thanks for your attention. Thanks.
So thank you very much, Professor Wu. The floor is open for questions. I might just ask, what is the connection between tumor cells and the Y1 peptide? I mean, is there any reason for that to be overexpressed or highly expressed in tumor cells?
Yeah, and the MPY, the ligand we usually use, for example, to use the covalent bonders attach the nanoparticle surface, attach the MPY molecular surface, the ligand on the surface. And because the tumor areas, for example, the breast cancer, there is very high expression rate for the Y1 receptor there. And they can get the attached on this tumor area membrane surface to get the internalization there.
I was wondering, why do you think the tumor cells express these receptors? Because they're neuropeptides. So why would they be expressed on tumors?
And these are a lot of literatures just before we do our work, and a lot of people just the literature showed in some tumor, for example, breast cancer, particularly the metastasis, the tumor breast cancers, the normal the Y2 subtype can transform to the Y1 subtype 100%. for the metastasis, the breast cancers, and also with very high just explosion rate there. And so after we searched this literature, so we decided to use the NPY1 receptors, design the ligand to get the targeting function there.
Okay. So, and also I would like to ask, do you know how much of the metals accumulate when you wanted to, let's say, make the MRI probes? How much metals accumulate? How highly are these receptors expressed? So how much would that give you? Did you actually measure the amount of the metal there? Because you need pretty high amounts of these to generate contrast for CT, for instance.
Mm-hmm. Yeah, very good question.
How much did you see how many micrograms or milligrams per milliliter or per gram tissue you would actually get?
Yeah. Okay, thank you. Usually for the clinic used the contrast agent for the MRI contrast agent, gadolinium is about the 10 milligrams about there. If you use the iron oxide, there is about 1 millimole the iron oxide there. For this one, based on the ions.
So that's using your targeting. You actually accumulate that much of your probe there to generate the contrast. That's a lot.
Yeah, and also it based on the just now I told you it's the amount usually for the clinic used for example for one person, for example the 60 kilogram rate, they use about 1 millimole the iron oxide nanoparticles for the get the imaging enhancement.
Yeah. So my question is, did you actually analyze the tissue from the mice to see the amount that was actually absorbed using ICP-MS or other techniques?
Yeah, we ever tested for use the ICP to test this one, and for in the tumor area in mice, rabbit, and also the mouse there. Yeah.
Okay.
Because we focus on the MRI contrast agent over 12 years to do this one, and also we are collaborating with the local pharmaceutical company to get the scale up there. Now we can produce, for example, about 1 kilograms for the iron oxide nanoparticles in the company. Because we want to get transformation to the products in clinic.
These are very impressive results, Professor Wu. This is really nice to see. I see that these are really could be clinically relevant of these compounds. I wish you luck in continuation of this. Is there anybody else from the audience that would like to ask questions? So if not, I think that we should continue with our panel.
Okay, thank you.
Thank you very much, Professor Wu.
Thank you.
Do we want to head the panel? I mean, is this you, Professor Gedenkin?
I don't know, but if nobody volunteers, I'll do the job. Okay, maybe I should start by asking Professor Ismach a question. What kind of application do you see for these 2D semiconducting materials? Or in other words, do you see any advantage? Because today semiconductors are all over various applications. Do you see any particular application to your structures?
Yes, so first of all, this is a very wide question because 2D materials, the family is a huge family which has superconductors, metals, semiconductors, and even insulators. So what if I find applications to each one of them? I don't know. And also to find applications, there is a trade-off between the properties and the quality of the material and then the price of processing it. But I think that these materials and also replacing the silicon technology, I think that many of us dropped that idea already a few years ago. But I think that these 2D materials are very interesting for a lot of applications that are related, for example, for flexible electronics and optoelectronics. And some of them, for example, hexagonal bond nitride that I didn't talk about it, but I do have some research on it, which is an insulator, but has very high thermal conductivity, which is usually not the case for insulators. So then these materials can be assembled into fields of thermal management. And we have a huge consortium, graphene consortium, trying to push graphene and hexagonal bond nitride to real applications in collaboration with the Mellanox, NVIDIA, and many other, Elbit and many other companies. So the idea is there, and graphene and hexagonal bond nitride, in this case, for thermal applications and high frequency devices can, they have advantages over traditional materials. And for the semiconductors, and again, these semiconductors that basically span all over the energy band gaps that are interesting for a lot of applications in nanoelectronics and optoelectronics. And also, if we manage to grow them, if we understand the growth mechanism and we manage to form, as I showed the schematic diagram, we can, in principle, make well-defined heterostructures that are only few atoms thick, but with many diverse properties in terms of energy band gaps and conductivity, metals, semiconductors, and so on. So this, I think, and all of these by methodologies that are not related to ultra-high vacuum processing. For example, in MBE, molecular beam epitaxy growth, you can grow very well-defined quantum well and quantum dot heterostructures, but you need for that UHV systems, which are very expensive in small areas and so on. So to push that technology to widespread is challenging, but these materials can be grown by low pressure or atmospheric pressure CVD processing, which is compatible with a lot of industries.
You showed, for example, the metal dichalcogonides. They are good tribological materials.
Right.
Did you ever try to use a surface coated with your material as far as to its tribological properties?
Well, we try to get funding for such studies on more complex materials like heterostructures of graphene and HBN. We have here in Tel Aviv University, Professor Oded Hod and Michael Orbach, who theoretically proved that these two materials, because of the very small mismatch between the lattice, they offer the possibility to have super lubrication, right? So we try to get funding to push this research into real applications, and so far we haven't, it wasn't so successful. But so these ideas are in the to-do list, but yes, we are busy with other.
Thank you. What about other questions? People, I urge you. Itamar, please.
Well, I think that for the discussion, I have two questions to two of the lecturers for this excellent lectures. One is directed towards Yuval Garini, and it relates to the perspectives. How do you see the perspectives of applying organized origami structures for solving biophysical problems, and what is your vision? In what directions should we go with the origami nanostructures to solve biophysical questions?
Okay, thank you. It's a tough question. It really needs a perspective of a panel. The way I see it, first of all, when we talk about research in biophysics, there is a lot to do. There was a very interesting discussion between me and Professor Dr. Eitan Lerner from the Hebrew University regarding different methods of using fret, but surface fret on metals that can extend the radius in which the fret can work. So I think for biophysical studies, there is a lot that can be done in order to study the structures of molecules, and it can get very far from where we are right now. Even maybe for studying the structure of proteins, the conformation of proteins, interaction of proteins, and so on. Because once you can label a nanostructure with different molecules, with different fluorochromes, or now make measurements of that in the nanometer and sub-nanometer regime, there is a very high potential. Another direction is in vivo studies. We already know, and we had even in a couple of lectures, that you can insert those some of those DNA origami structures into live cells, whether you do it through liposomes or even as a standalone structure, once you add to them the right sequence that can penetrate into the cell, maybe even into the nucleus. Now, just imagine, for example, that you make a DNA origami that can penetrate into the cell, dissolve in the sense that the structure disassemble, and suddenly you have DNA. This DNA may be used either as a code for synthesizing protein or as an siRNA in order to interfere with what's happening in the cell. And the direction from this toward what is being done today with CRISPR, for example, is, I think, very, very short. And there are even few works that are done, I don't remember the exact scientists, but at the National Center in Beijing. So that's a second. The third one that I know much less about is in the direction of the last lectures that we heard on using those as vehicles for introducing drugs into cells in a very precise manner, in a very targeted manner under what's called the precision medicine.
Excellent. Very good. The next question is directed to Professor Wu. We are witnessing in the last 5-10 years a lot of studies that are related to the application of MOF nanoparticles, metal organic framework, for imaging, drug delivery, photodynamic therapy, and so on. My question is, and I'm not familiar to what is done in China, but I would like, if you can comment, is there any one step further in China already? Do you know something about approval of these particles for some practical cancer therapy or diagnostics? Do you know about it?
I try to answer your question, Professor Eitan Wiener, because I'm the student from the Professor Kang Wang. So he's my PhD supervisors. And also I know you for over 20 years and admire you so much. And so I try to answer your question. In China, now they combine the morph-based structures with the, just now you mentioned imaging and the therapeutic unit there. And recently, some Chinese group use the morph and attach some the catalytic activity atoms and to get the enzyme like the therapeutic function there. And I think these are some the new recent work for this area, for the morph there. But for us, for our work, just we use the morph to do the experiment. Just want to seek for some basic questions. If you want to go to the clinical applications, I think there is a different kind of the things to show the work, because for the sciences, we try to get very novel and very definite things to do. But for the clinical use, the products, they try to get the very simple, because for the pharmaceutical, the consistency is the first thing they want to try to resolve it. So in my opinion, if they go to the clinical use of the products, and also for the basic sciences, we should have a compromise for those ones. This is my idea. Maybe my opinion, maybe not good, but this after I just do the experiment in the nanosciences for the biomedicine application. This is my idea for this one. So I don't know whether I can answer your question or not.
Okay, thank you.
See if anybody else has an answer to your question. For example, does anybody knows what kinds of regulation problems we will face once the morphs will come to approval of the FDA or the European Community? Anybody else besides Professor Wu wants to answer this question? If not, let's go to the next question. Who else wants to ask? Professor White, please. Professor White, go ahead.
Yeah, I'm just unmute. Yeah, so yeah, I have two short questions. One is for Professor Ariel Asma. Yeah, actually, the growth of the two-dimensional semiconductors in large area is a big challenge. But graphene as beginning also faced a similar challenge, and now people can doing some really nice large area graphene layers on the copper single crystals. So what will be the solution for two-dimensional semiconductors to get large area uniform controlled layers two-dimensional semiconductors?
I'm sorry, I guess that I missed part of the question. So what is my solution for large area growth of 2D materials?
Yeah.
So as I try to emphasize in my talk with all the problems that I had in the presentation, I try to emphasize that one of the ideal cases would be to be able to grow these materials epitaxially on a substrate, right? This is like the state-of-the-art technology semiconductor industry in which we grow epitaxial thin films of silicon germanium, and gallium arsenide, of course, and many others. Now again, these materials are layered compounds, so van der Waals epitaxy is kind of elusive, and we are trying to dig into the exact mechanism, which might be more complicated than the standard description of just van der Waals formation on top of a crystal. But we're getting there. So, so I think that I'm actually, and this is also my next few sentences are connected also to the first question by Professor Gedanken. I'm really optimistic about these 2D materials reaching real applications. Maybe not all of them, but some of them they will do. And one of the reasons for that is that all the technologies that are built today, the high-end technologies, are built on 2D technologies. And these materials are the ultimate 2D. These are 1 to 5 atom, usually thick films, crystalline films. And I think that if we are able to solve this bottleneck of understanding the growth mechanism of this film and to push further for the growth on large areas, as we are aiming to do, I think that I'm very optimistic about the implementation of such materials into real applications. But you need to take into account that when you have a new material, it might take a few decades from the lab to the industry. You know, the first time that the group in UT Austin that I joined for my postdoc developed this CBD methodology to grow a large-scale single-layer graphene. That was in 2009. And only now and few years ago, industries are pushing the same methodology into real large scale. But it works. And of course, there are a lot of questions and a lot of implementation and optimization, but it works. So that proves that once you understand the growth mechanism, and it took some time to understand and study the kinetics and the thermodynamics of the growth, it took some time, but once you do understand the growth mechanism, it is possible to push that forward.
Yeah, great. Yeah, so my second question is to Professor Yuval Garani. Yeah, actually DNA nanotechnology is really beautiful. Yeah, actually recently in your center, Professor Baoquan Ding showing some application using this kind of DNA origami as drug delivery applications. But the cost, the price of this DNA origami always quite expensive at this stage. Yeah, so it's possible in the future to overcome this upscale problem for DNA origami?
Yeah, well, it's not something that I was working on, but as you mentioned, there are actually already some more novel ways of designing and structuring DNA origami. For example, without using the scaffold, just by using very short oligonucleotide sequences, and the price of those short oligo sequences is dropping down constantly. And I can even imagine that once it will become there will be a real product, a real need for that. I'm sure that there will be ways to produce those in massive scales, for example, just like we do with plasmids in bacteria. And then you can reduce the cost enormously, not just by growing oligonucleotides, one nucleotide by the other. So I think once there will be a need, the cost will drop down significantly. I mean, look at what happened to DNA sequencing. It cost $10 billion for the first human sequence, I think, they estimated, something like that. The price reduced to something which is on the order of $1,000 to a few $1,000s per person nowadays. And there are already companies working on having sequencing for something like $100 US, with companies that very soon will have the product. The same if you look at the price of oligonucleotides, it's dropping down enormously. So I think the potential will be there. Once there'll be the real product, whether it is for drug delivery or for gene editing or whatever, we will see. I mean, look what happened now with the COVID-19. The cost of those tests, it was and the time it takes, by the way. Without even thinking, in 1 year, the costs went down, the time for the diagnostic test went down. So I think the same will happen with DNA origami.
Well, I must say that this optimistic view of Professor Garani is not reflected in my expenses, in my research expenses. I would like to have him as my accountant to be a budget leader. In any case, but to answer also Professor Weiss' question, I think that William Shih at Harvard, he has some idea, and he has even some published literature on trying to solve and to address this question by making the origami using a PCR approach. So this can make it much more economical and cost-effective. So there is something. I think that if I can ask my moderator and chairman, we have usually time restrictions here. And if they want to say something in addition and to push towards heading a question, go ahead. If this is not the case, I think that we should let our colleagues from China to try and to find a nap and sleep. Because one of my duties is at the end of this webinar to have some concluding remarks. And
I'm expecting from you concluding remarks.
You expect them. Okay, so if this is the case, first of all, I would like to thank very much the speakers of today, the chairman, the moderator. All of you made an excellent contribution, and it just added another value to the previous two webinars, that at least the echoes of them were very positive from China as well as from Israel. So I think that our meeting today is totally positive to this contribution. I think that what we have heard here today, and this was really, nanotechnology has a very bright future, and it is, I think that we should have more and more and more discussions in this area. And the identification of the link today between biomaterials on one side and self-assembly from the other side was reflected extremely nicely by these wonderful talks on medical application, diagnostic application, and the future of nanostructures for medical uses from one side, and from the other side, to go from to contribute the self-assembly to semiconductor industry, to semiconductor and photonic and optical electronic properties of semiconductors. I can only say that this combination and this blend has only emphasized, first of all, the significance of basic research. And I'm very happy that all of you valued the significance of basic research, but at the same time, you emphasized the practical utility and the future possible applications and results of this basic research. And I think that with these comments, we have a wonderful, I think, concept for the next webinar that will address medical and medicinal application of nanotechnology. And the talks today are an excellent bridge from what we had in the first two seminars to the last fourth webinar. I can only hope that all of us will have the opportunity to visit soon China. At the moment it doesn't look so bright, because any scientist has to enter into a quarantine of about 2 weeks. And I don't think that many of us can survive under this restriction. But let's hope that soon I can be, at least I can be again, a frequent visitor to China. So I think that I should thank all of you. I would like to thank all the infrastructure of the Chinese Academy of Science and the Israel Academy of Science. It was a wonderful webinar, and I look forward to the last one. Thank you, all of you.
Thank you as well. Bye-bye.
Bye.
Bye.
See you. Bye-bye.